Hello everyone. My name is Chen. I'm currently a R&D manager at Sino Biological. Today we're going to talk about recombinant antibodies, what they are, and how are they made. So I would like to structure my seminar into these four parts. So first I would like to give a quick overview of our company, and then we're going to move on to talk about recombinant antibodies, their derivatives, and the strategies involved in their expression. And last but not least, I would like to quickly touch bases on a novel high-throughput expression platforms that we have established to help create antibody libraries. So, company overview. Sino Biological was founded in 2007. Our headquarters is currently situated in Beijing, China. And as you can see from this map, we have offices in North America, Europe, Japan, and also some other locations in China. So basically, we're everywhere. The company has more than 500 employees at this moment, and our facilities has been certified by both the domestic as well as international agencies. Our core business involves reagent and custom services. Our reagent ranging from genes, proteins, antibodies, as well as ELISA kits. So we have created a significant library of these biochemical reagents to facilitate biomedical research as well as drug development. So our products are used by researchers in over 90 countries, and I think they're well received, as you can see from this citation charts, which is on an upward trajectory. And besides our product pipeline, we also offer contracted research services to research institutes as well as biopharmaceutical companies. So what we offer mainly is to use our recombinant expression platform to help you prepare recombinant proteins as well as recombinant antibodies, which you will hear a lot during this talk. And we also have an antibody generation platform that we use different techniques such as mouse hybridoma or other library construction methods such as phage display or a single B-cell sorting technique to help identify antibodies of desired functionalities. So that was the quick introduction of Sino Biological, and if you're interested in our company, I would encourage you to visit our website. And now let's move on to our main course of the day, which is to talk about antibodies and their derivatives, what they are, and then we will talk about strategies of how to produce these antibody constructs using recombinant expression. So I'm a protein biochemist and not an immunologist, so I would forgo this the detailed explaining of this chart of how antibodies are made. But essentially, antibodies are secreted by matured B cells. They're essentially sentinel molecules circulating in our bloodstream. They can interact with a particular on a particular part of an antigen with high specificity as well as strong affinity. And so these are the features that actually make the antibodies very appealing and bear the name of a magic bullet. And that is also why they're I think one of the major reasons why they're the focus of a lot of pharmaceutical companies, and I think they're considered as the foundation of modern biopharmaceuticals. So in our body, there are in our blood circulation, there are 5 different types of 5 different subclasses of antibodies. They're mainly categorized by the chemical nature of their heavy chain. Antibodies do share light chains, which comes in either a kappa formation or a lambda formation. Okay. So among these 5 antibody subtypes. IgG is the most abundant, followed by IgM and IgA, and we only have a small amount of IgD and IgEs in our circulating our bloodstream. So for the IgG format, it can be further categorized based on the sequence of the constant region as well as the length of the hinge region into these four subtypes. And among which I think IgG1 and IgG4 are the main formats that are used in biopharmaceuticals. And we also have unique formats of antibodies. So, for instance, llama and other camels can generate single domain antibodies, which is comprised of only one heavy chain. And there are antibodies called IgY in the egg yolk, which has a rather complicated Fc domain. So I think the field of immunology and antibody can be traced all the way back into the 1700s, and this field has been developed for over 300 years. But I think up to 1975, when the hybridoma technology was invented, this field took a exponential growth and create and generate more and more therapeutic antibodies that adds to the arsenal to fight either cancer, infectious diseases, as well as other type of diseases such as autoimmune disorders as well. So the development of every new piece of technology resulted in the addition of novel antibodies into the pipeline. I think up to 2022, up to this year, there are over 135 antibody therapeutics. They're either approved or in the regulatory review stage. And with the rise of artificial intelligence, I think there will be more and more antibodies to be developed and approved in the future. And among these previously proved antibodies, you can see several of them are among the top 10 drugs in terms of their global sale by 2019. So it's not only the antibody development. It's not only a technological challenge, but it is also a highly rewarded business. So I think that's why a lot of big pharmaceutical companies are invested heavily into this field. So, like I mentioned earlier, antibodies are actually very specific and highly selective to their specific targets. So that determines that they can be used either as therapeutics, like I mentioned earlier, or as core reagents in diagnostic assay tools. And also they're really valuable tools for biomedical research and can be used in different type of immunological assays such as Western blot, immunofluorescence, immunohistochemistry, and so on. These antibodies help us either quantify or track the location of the target that we're interested in. And here I would like to also take this opportunity to give a quick shout out of this new anti-Claudin 18.2 antibody that we have developed recently. It is a immunohistochemistry applicable antibody, and it outperforms I think the current versions of the commercial Claudin 18.2 antibodies as well. So if you're interested, I think you can request for a small sample from us, and I will have the contact information on the last slide of this presentation. So keep an eye out for that. So antibodies are useful. They can do a lot of things, but they all bear several key regions that carries out these functions. One of the most, I would say, the most important region would be the variable region. It is comprised by a combination of 6 complementary determined regions, CDRs, on both heavy chain and light chain. These CDRs forms this paratope that interacts with the epitopes on an antigen. And The Fc domain, of course, also carries out its biological functions, which uh, we will mention later. Uh, but everything actually starts with a sequence of the variable regions. Once you have that, we can do a lot of engineering, as well as and to convert a full-length IgG into different formats. So depending on where you obtain your variable region sequence, so if your antibody was derived from a hybridoma culture, you can take the cells, extract the mRNA encoding the antibody sequence, and then we can perform next generation sequencing and piece together the sequence of the CDR regions to generate this to decode this variable region. But if you were using a other type of discovery process, for instance, phage display or B cell sorting, then you will have after the panning process, you will have sequences to begin with, so that will make things a lot easier, well, somehow easier. So once we have the amino acid sequence or the gene sequence of the antibody, the variable region available, that we can convert them into different formats. I listed a few formats here. These I think are the most common formats of recombinant antibodies. So the first one is of course fragmented antibodies, which means we can take the variable region and try to miniaturize them into either the Fab or a single chain FV version of the antibody. Uh, and we can also graft the variable region of one antibody onto the framework of a second antibody. And we can also fuse scFv or VHH, these smaller antibody variants onto a FC to enhance their serum stability. And lastly, I would like to mention with the variable regions of one antibody, two antibody, or so even a few antibodies, we can try to put them together and to create bispecific or trispecific or multi-specific antibodies using protein engineering technique. So here I listed several formats of bispecific antibodies that we have encountered during the years from our customer services. But for a more comprehensive review of what bispecific antibodies are and what kind of formats they're in, this is actually a very good review. I've listed the reference here. If you're interested, you should definitely check out this review paper. Now we've touched briefly on the formats of different antibodies and know a little bit about where they come from. And now I would like to spend some time talk about how we can create these different formats of antibodies using recombinant expression, the recombinant expression technique. So since we're trying to make a recombinant version of your antibody, so it still adapts to the whole recombinant protein expression, central dogma, if you will. So basically, we take a target gene, in this case, the gene or genes that encodes your antibody of choice, and we insert them into an expression vector, and then we figure out a way to transfer these expression vectors into a host cell, and we let the host cells do its magic to create a crude protein or antibody preparation, and then we can extract the antibody of interest from the mixture using purification techniques. So common host cells used for recombinant protein expressions are listed here. Each of them have their characteristics and they have their pros and cons, if you will. And we routinely use all these platforms for recombinant protein expression. But in order to make a recombinant antibody, the first thing that we need to do is to select a suitable host to do this job, because the host cells are very important, so that the selection of a suitable host will largely impact the success of your recombinant either protein or antibody expression project. So, in order to select the host cell or the most host cell for recombinant antibodies, we would need to take a closer look at the antibody molecules. So first of all, they're secreted proteins, which means they need to be uh, uh, secreted out into the culture media in order to be purified. And antibodies themselves have very complex high-order structures, and they have multiple disulfide bonds in the sequence, not only in the hinge region but also in the constant region as well. So we need to make sure that they're correctly formed, and the correct folding of both the variable region and the constant region are important for its functionalities. So the corrected folded variable region is crucial for antigen interaction, while that of the Fc region is important for the Fc receptor interaction as well as its downstream effector functions. And antibodies are glycomolecules, and they bear glycans that are important for the stability and their functionality. So we would want these glycans to be present in the mature molecule as well. So to put all these factors together, it is sensible to choose mammalian cell systems such as HEC293 or CHO cells to serve as the expression host for recombinant antibodies. However, I think in the literature, there are reports indicating you can use either E. coli or yeast to serve as hosts for the expression of especially VHH type of antibodies. So here I listed a reference here, which I think the authors took advantage of a HLY secretion system of E. coli, and they created this, secreted form of VHH fusion protein. So this is one way to produce VHH in E. coli. But I think the majority of the recombinant proteins recombinant antibodies, are produced using mammalian cell culture systems. So once we have the antibody sequence, it can be derived from antibody discovery campaigns from using different techniques. Once we have the sequence, we can put them into expression vector and carry out a small scale or transient expression experiment and collect several antibodies to test. And once we determine which one or ones we would like to proceed to a larger scale manufacturing, we can then try to create stable cell lines and to optimize the culture and purification protocols to achieve larger scale manufacturing. In this talk, I will not cover the discovery part as well as the stable cell line part. There can be stories for another time. But rather, I would like to focus on this central process that starts with the antibody sequences and all the way to antibody purification and activity assessment. So briefly, once the antibodies or the sequences are obtained, they're cloned into different expression vectors. And usually, if you want to express a full-length antibody, we use two vectors. But depending on the actual format of the antibody, it can be two vectors, one vector, or sometimes even more than two vectors. And the vectors are transfected into the host cells using this PEI-mediated transfection, and the cells harboring the expression vectors undergo culture in different scales, from culture plates all the way to those giant the bioreactors with giant volumes. And at the end of the day, the antibodies are purified, and assessed for the activity using appropriate tests. So I would like to break this core process down to its different parts and talk about them separately. So first, I would like to talk about the initial step in this process, which after obtaining the antibodies is what we can do in terms of construct design to make sure that we will be able to produce a fully functional antibody. So the antibody molecule contains the variable region, constant region, which also bears two major sites of glycan. So because they're secreted molecules, so we would have to add signal peptides to the N-terminus of both the light chain and the heavy chain to facilitate the secretion. And with the variable region, we can produce a full-length antibody, of course, but we can also just take that part and make it into a smaller version, a process we call the miniaturization. So we can generate single-chain SCFVs for it from this part. And there are several factors that I would like to discuss in terms of a SCFV construct. And the antibody, like I mentioned earlier, is a glycoprotein, so the glycan can impact its functionalities and can be modulated. So we will take a look at these three different aspects in terms of the antibody construct on this slide. So first of all, signal peptides. So here is a list of signal peptides that we use quite often in terms of antibody and protein expression. The ones highlighted in red are commonly used for antibody expression. Please note that these signal peptides are also suitable for expression of SCFVs or VHH antibodies and so on. And they do have an impact on the yield of antibody, as indicated in this study, where I think the authors in the first graph they did I think they did a combination of different signal peptides on the heavy chain and light chain, and they identified a combination that resulted in about 2.5-fold increase in the antibody yield. And on the bottom chart, I think they did a scan, did introduce the mutations to the signal peptide on the heavy chain to identify which residues are crucial for crucial for antibody secretion enhancement. And another thing to remember is if you were trying to express a protein antibody fusion, then you might want to choose a signal peptide to help the secretion of this molecule. And so here's the listed website, Signal P6, I think it's 6.0 now. This is a very useful website to help determine the cleavage potential of the signal peptide that you choose. Now let's circle to the glycan part of the antibody. So the core fucose can modulate the ADCC functionalities of antibodies. So I think one way to enhance ADCC function is to remove this fucose residues, and this can be achieved using fucose 8 double knockout 8 HEC293 or CHO cell lines. The antibody is expressed using regular HEC293 or the fucose 8 double knockout. HEC293 doesn't show too much of a difference in terms of their SEC profile. And however, if we examine the molecular the molecules a little bit more in a little bit more detail using mass spec, we can see the difference in their molecular weights. So this is so the FUT8 double knockout cell lines is the way to achieve a fucosylated antibody and for ADCC enhancement. Now let's come to the miniaturization process. So in order to convert a variable regions of the antibody into a scFv, there are several things that we would need to consider. So first is the linker, what kind of linker we want to use, and the length of the linker as well, and what kind of purification tags that we would like to put on this construct. And sometimes we also would have to add some accessories to facilitate other type of functionalities, which I'll show in a case a little bit later. So in this paper, the authors investigated the length of the linker and to identify their impact of the activity of the scFv molecule. And I think what they found is that the linker has to exceed certain lengths. So for instance, 15 amino acids, 3 repeats of this GGGS linker should adequately ensure the functionality of the scFv, while if the linker is too short, then sometimes the functions of the molecule can be somehow compromised. And here is an example of one of our previous projects, which I would like to emphasize that we will touch bases on these accessories. So for instance, the goal of this project was to express a scFv his tagged scFv with an AVI tag, and to achieve in vivo biotinylation. But the first construct we produced is a low yield with low biotinylation ratio, and it showed a great extent of aggregations. So as in the optimization, we added a spacer between the his tag and AVI tag, which you can see improved the yield about 10-fold, 12-fold, and also improve the biotinylation ratio as well, and reduce the aggregation to only about 10% oligomer. So the spacer does help stabilize our molecule. And the last and another attempt is that we replace the his tag and the spacer with an FC tag in order to put more space between the scFv and the AVI tag. And the yield improved more significantly to larger than 25 mg per liter. And also the biotinylation ratio is secured at 0.9, and we only observed about like 5% aggregations. So what kind of tags you put and how to rearrange these tags, how to arrange these tags, or if you would like to insert any other accessories between the tags, can indeed impact the final performance of the molecule that you produce. So we would encourage to design multiple constructs in order to find one or a few that can result in a most stable molecule with decent yield. So there are several things we can do to the construct of the molecule of the antibody to facilitate its expression, but we can also introduce modifications to the Fc domain of the antibody to either enhance its stability or add on additional functionalities. I'll show a few cases on this slide regarding Fc modifications. So the first case is about to introduce cysteine mutations so that we will have some orthogonal conjugation sites on the Fc domain. So I think what the authors did was they scanned the spatial aggregation propensity of the Fc domain and identified a site on the CH3 region that has the low. I think that has a relatively low potential for for protein aggregation. Mutated that to cysteine residue, and which resulted in this V10 construct, which this construct expressed at a comparable yield to compared to the wild type, and it also had a good monomeric content in the antibody product, which is important because sometimes the addition of cysteine residues can cause unwanted aggregations, and this uh, antibody can be selectively labeled at the CH3 region by their fluorescent assays. So this is one example of how we can modify FC region to insert or install some conjugation sites here to achieve site-specific conjugations. While FC is also modifications on FC is also important to enhance the stability of this either antibody or FC fusion molecules, as one example shown here. The story about this case is that people find um, there are some indigenous instabilities in some antibodies or FC fusion proteins, which will result in the cleavage of the FC tag, as indicated by the arrow here. After long storage, they discovered that the FC tag kind of falls off, at least some of them. They contributed this phenomenon to the atmosphere. Oxygen gets involved and creates hydrogen peroxide, and which attacks the hinge region and result in this cleavage. So what they did to remedy this issue is they introduced a mutation into the hinge region, and upon the introduction of this mutation, this problem was greatly was somehow resolved, and the the degradation of FC was successfully prevented. And we also did a similar trial ourselves. So we expressed a FC fusion protein. We introduced this substitution, and as you can see, for the the wild type FC, there are about 30% FC cleavage, but this after the introduction of this mutation, the FC cleavage was significantly reduced, and this mutation was not and does not impact in for the FC in terms of its functionality, at least in the Western blot assay. So this is a mutation that worth considering if you do observe, the fall off of the FC tag in your construct. All right, last case would be to introduce cysteine pairs in the FC region that forms intramolecular disulfide bonds to solidify this region so that we can have an enhanced stability of the construct. So in this paper, the authors introduced actually two pairs of cysteine residues into either the CH2 domain or the CH3 domain. The antibodies were made, and they were actually the FC fusion proteins were made, and at similar expression level, and they showed similar functionality, similar secondary structures, judging by the CD spectra, as well as a similar interaction with the corresponding FC receptors. However, the difference shows when these molecules were subjected to heating. So once they were heated up to 60 degrees, you can see that that FC domains with this CH3 pairs disulfide bonds exerted this extended stability comparing to the wild type as well as the one with only the CH2 cysteine pairs. So this is also a useful technique to help improve the stability of your antibody or your FC fusion proteins. Now, once we have the constructs all settled, then the next thing we would need to consider is what kind of a culture conditions that we would need to subject our own constructs too, in order to have an optimized outcome in terms of their expression. I think the first thing would be the expression vectors. So every company, I think, will have their own optimized vectors to go with their transfection reagent as well as their culture media. Uh, so we, Sino Biological, we have also our proprietary um, expression vectors, which you can see it outperforms some commercially available vectors in terms of antibody yield. So once the constructs are inserted into the expression vectors, the expression vectors were then transfected into host cells by PEI-mediated transfection. So PEIs are essentially these positively charged polymers that are used to encapsulate the negatively charged DNA molecules, which are the plasmids, and help to deliver the plasmid into the host cells and more preferably into the host cell nucleus. A efficient transfection reagent will greatly help and ensure the success of your recombinant antibody expression work. So you would want to select one that works best for your system. And then it comes to the culture conditions. So of course, culture media plays a significant role in this process, but also there are other parameters that we can make adjustment to. So for instance, the culture duration, the culture temperature, and some additives that can also be added into the culture to boost the protein yield or to reduce protein aggregation or degradation and so on. So here I would like to actually show you a case about our recent campaign in order to look for a best combination of transfection reagent and culture media to enhance antibody expression. So for this work, we selected 20 low expression antibodies whose expression levels were below 20 milligrams per liter. And we optimized by taking two culture media recipes, M1, which is the ones that we're currently using right now, and M2, which is a modified version based on M1. And we also took two transfection reagent formulas, T1 and T2. T1 is the original version. T2 is the updated version. So we kept the culture volume the same, the duration, and the temperature. We kept all these parameters the same, and we just want to investigate what kind of impact of the combination of culture media and transfection reagent will have on the yield of these 20 low expression antibodies. So the results shown here, you can see clearly that the combination of M2 and T2, which are the updated version, they do result in the increase in antibody yield for all tested antibodies. And we then took a closer look and tried to find a correlation between transfection efficiency and antibody yield. And we, in this GFP assay, and you can, it's depicted here. You can see that when we use this M2T2 combination, we observed a significantly more strong GFP signaled cells when compared to the M1T2 formulate M1T2 combination, which actually it indicates the more efficient the transfection, the higher yield you will have for your recombinant antibody. So I think the take-home message on this slide is you would want to optimize the culture system, and especially the choice of transfection reagent and the culture media in order to achieve a maximum yield of your recombinant antibody. Now, once the antibodies are cultured, I think the last step will be to purify these antibodies from their culture media. So the purification will follow this scheme. We have the crude sample. We try to capture the antibodies somehow, and then we polish them using different methods. But lucky for us, antibodies have the Fc region, and the Fc regions are considered as a quote unquote endogenous affinity purification tag. So there are different proteins, especially protein A and protein G, derived from bacteria. They specifically recognize the Fc region, and these proteins are routinely used to serve as affinity purification tag for antibodies or Fc fusion proteins. And for some antibodies, if their light chain is in the kappa format, then another affinity approach, protein L, can also be used to selectively purify these antibodies. However, if you have only like a fragment of the antibody, or your antibodies contain some mutations that prevents the interaction with protein A or protein G, then we can simply just treat the antibody as a recombinant protein and purify it using a combination of chromatography methods. So here I would like just to give a quick shout out to this web tool. It's called the PI calculator. It can help you, calculate the PI or molecular weight and other, parameters. By introducing, subunits, so which is kind of useful to figure out what kind of to figure out the physical status of the antibody of interest, and plan your purification work accordingly. So here I would like to showcase a previous study that we did. We made a bispecific antibody for a client, and the first step of protein A purification did capture the majority of the antibody. However, we do observe some content in the format of a oligomer, and then we performed two consecutive steps of purification, an ion exchange, as well as a gel filtration. And after these two steps, we reduced the oligomer content to about less than 5%. So I think the take-home message for this slide is that antibodies are relatively easy to work with because of their affinity tag, but sometimes other purification steps are required in order to refine the quality of your antibodies. Okay, I think on the previous slides, and I think what we were talking about are either full-length IgG type of antibodies or fragmented antibodies. However, IgMs I think they're becoming more and more trendy these days, because they're either a pentamer or a hexamer, and because there are multiple valencies, I think people, especially I think in the IVD industry, are paying more and more attention to these formats of antibodies. Because of their multiple avidities, it could increase the sensitivity of their assay. And IgMs can also be produced using recombinant expression technique. So for the pentamer format, which means that it has a J chain to help facilitate the formation of the pentamer, we can normally use two transfection methods to obtain a pure IgM in the correct pentamer format. And also the hexamer IgM without the J chain can also be achieved as well using similar approaches. However, I would like to mention that because of their size and their oligomeric status, the purification of IgMs could be a little bit trickier. So if you have an IgM and it has a kappa light chain, then we can use protein L affinity to serve as the first step of purification to help capture the antibody, and then we can use a second purification step. I think in this case we use the gel filtration to remove the smaller molecular contaminants and to enrich the pentamer and the pentameric and the pentameric format of the antibody. However, if your antibody does not have a kappa light chain, then of course there are purification matrices available that specifically recognize IgM framework, but they could be somehow costly. So for us, what we do is we treat in IgM as a recombinant protein. So we use a combination of different chromatography steps to obtain a purified version of the molecule. So I think in this case we took two-step approach. The first is an ion exchange, followed by a gel filtration, and you can see after the two steps we can also purify an antibody with larger than 95% purity. So the take home is IgM can also be produced using similar approaches, but their purification may be a little bit trickier, because of their sizes. Okay, lastly, I would like also to take this opportunity to give you guys a brief introduction and a brief quick overview to our high throughput antibody or high throughput protein expression platform. So how it works is that we can collect an antibody sequence library from a client, and we design primers according to the sequences of the antibody, and then we will subject these design to high throughput primer and primer synthesize and vector synthesize. So these are kind of automated and process, and once we have these vectors, we transfect a array of HEC293 cell culture in the format of shaker flasks. And at the end of the culture duration, we used a protein A affinity column to perform a one-step purification. Normally, we can get up to 90% pure antibodies, and we can then subject them to function validations, in this case an ELISA assay, and select a few ones with the desired functionalities and go to the scale up process. So we use this shaker flask platform because the yield results from this culture method is more relatable to the final scale up. We have performed actually several projects using this platform, and we have generated over, I think, 6,000 antibodies, I think, to this day. So I want to show you two cases. So the first one is we helped a group to create a library of antibodies against COVID-19, and we expressed these antibodies full length human IgG1, I believe, using this platform, and our collaborators identified 14 neutralizing antibodies within this library, and we scaled up the production and generated 10 to 300 milligrams of the material, and they used these antibodies for structure analyses and animal studies, and the the results were published in Cell in 2020. And not long ago, we also did a customer VHH antibody library generation. The size is a little bit smaller. We uh, made 74 antibodies, all in the format of VHH FC fusion, and all the antibodies are produced successfully. And after one-step purification, we get a average yield about 50 mg per liter, sorry, and also the SEC monomeric purity at about 95%. So this system has been proven to be quite robust and efficient in produce both full-length IgG antibodies as well as VHH, the VHH variant. Okay, to sum up, so antibodies are essential molecules. They're a very they're very versatilely they have versatile functions, and they can be used in a lot of research areas in the life science realm, and they can be derived from various methods, and the massive production is usually achieved by recombinant expression, which was the main topic of our discussion today. And they can be engineered into different formats, and we prefer to use mammalian cell lines as expression host. And the antibody and the expression of an antibody can be optimized at different steps during the process, from the initial construct design all the way to the selection of the most suitable culture methods and purification strategies. And of course, this high throughput platform is quite powerful, and it enables the production of both full length as well as VHH fusion format of antibodies. And we would hope to using this platform more and more in the future to help clients create their antibody libraries. So with that, I would like to conclude this presentation. Thank you for taking the time to attend our seminar. And here are our contact information. You can reach us either by phone or by email, or by scanning this QR code. With that, again, thank you for your time, and I would be happy to take any questions.